Chinyenze, and I work with IAVI, where I serve as the Executive Medical Director. I lead the clinical development team that is based in the Africa region to conduct clinical trials and epidemiological studies in the region with partners that are spread across 5 countries in East and Southern Africa. My background is I'm a physician, and I have a passion to develop HIV vaccines that will ultimately be deployed for populations in need across the Africa region. So today I'm just going to give you a quick snapshot of the current HIV vaccine pipeline. And we will be focusing on the current efficacy trials and looking at some of the products that are in the early stages of development. It's always good to remind us how the HIV pandemic continues to be an important and significant global issue, with 75 million people having been infected since the beginning of the pandemic, and 1.7 million being newly infected in 2019. This is an issue of great concern. If you look at about 5,000 people becoming infected each day. And when you look at the data more closely, we acknowledge that there are certain populations that are being left behind. We see that key populations account for about 62% of new HIV infections worldwide. And this is unacceptable in 2019, so many years after all these prevention efforts. And similarly, in adolescent girls and young women, continuing to account for up to 25% of infections in sub-Saharan Africa. We have made a great deal of progress in the rollout of antiretroviral drugs for therapy. Unfortunately, we also continue to see up to a third of people who are living with HIV in 2019 not being able to access treatment. There are existing prevention tools and what we tend to refer to as HIV prevention toolbox, a range of tools targeting different populations at different times. And just to highlight a few of them, when we look at PrEP, it has, whilst it has worked in certain parts of the world, we are seeing some challenges. For example, in Kenya, that was one of the countries that was leading in the rollout and of PrEP. What we are seeing is low levels of uptake of PrEP and low and poor adherence in populations. We are seeing challenges in certain populations such as female sex workers or MSM and transgender individuals. And some of these are related to issues such as stigma related to the taking of this drug, which is also similar to the drugs that are used for HIV treatment. There are also concerns around the side effects. And really, I think one of the major challenges is the requirement for daily adherence to this oral tablet. And when you look at treatment as prevention, we also know that it's very effectively antiretroviral drugs suppress the virus and reduce further transmission. But again, there continue to be challenges around early identification of people who are infected, getting them on treatment and having them stay on the treatment for life. And also side effects are challenging for these groups as well. When we look at the incidence at a global level, there has been significant progress over time. And this has been due to the implementation of the broad range of prevention tools that we referred to earlier. Unfortunately, the incidence continues to be unrelenting in that we don't see progress towards the goals that had been set for 2020. So 2020 has come and gone. And unfortunately, we did not reach the target of 500,000 infections by the end of 2020. And we're waiting for the data for UNAIDS to come up. But one thing we are concerned about is the impact of COVID that is had additionally on incidents. UNAIDS estimates up to about 148,000 more people could die from HIV/AIDS due to disruptions in health services. And similarly, we expect to see disruptions in prevention services. And so this may also have impact on incidents going forward. And all this really highlights the continued need for an HIV vaccine. However, several decades later, in the development of an HIV vaccine, several scientific challenges have continued to plague the field. And some of them are not new. They've known to many. HIV is an incredibly hypervariable retrovirus. It has a lot of genetic diversity, and it has a lot of effective mechanisms of immune invasion, for example, the glycan shield that it deploys, as well as the flexibility of the envelope. And all these combined with the genetic diversity make the HIV virus a formidable virus in terms of trying to have the immune system respond and effectively control the virus. There's also a lack of ideal animal models for predicting vaccine efficacy, as well as no proven correlates of protection that facilitate the selection of vaccine candidates with a high probability of success. And one important thing to also note is this growing range of HIV prevention tools is really making efficacy testing in human populations increasingly more complex. There's a potential for much larger trials that may be necessitated in the context of these prevention tools, making research more expensive, and also the designs really complex. But there are some exciting things that have happened recently that do offer hope and progress that has been made and is showing us that there are opportunities that lie ahead, and the path to developing an HIV vaccine continues to be feasible. And just to highlight a few of these, there has been the isolation of broadly neutralizing antibodies from human samples. We shall talk about this a bit more, and how this has contributed to the identification of epitopes as potential targets for vaccine design, the advances on the mRNA sort of vector technology front, again, another area that offers a path to develop and iterate vaccine candidates faster. There's also a rich pipeline of products in early and late phase testing, and we will talk about this starting off with the current vaccine trial, efficacy trials that are ongoing, and then we'll go on to the experimental medicine trials. So this, just to give you an overview, there are currently 3 trials that are currently ongoing. The Invocado study, which is a phase 2B trial, and the Mosaico study, which is a phase 3 trial. Both of these are a part of the Janssen's Ad26 HIV vaccine program that covers a range of strains where this vaccine is designed to be able to cover the multiple strains that are found globally. And then the Prepfact trial, which evaluates the efficacy of 2 vaccine regimens in the context of oral PrEP. And so the next few slides, we're going to talk about these studies, starting off with the Invocado study, Invocado trial. So as we mentioned, so this is the Janssen vaccine that is designed to provide cross-clade protection of HIV, deploying the Ad26 vector, which was chosen for its low seroprevalence, and also looking at mosaic inserts that were selected for their global coverage, and also adding GP140 covering clade C. And so this trial has already started and is actually in the middle of implementation. The goal was to enroll about 2,600 participants. They have just reached that goal, just slightly past that target, and completed enrollment. So this trial is actually giving 4 injections over a space of 12 months with a follow-up period of 36 months. And this is more of a proof of concept trial. It's a phase 2B trial to see if the vaccine will work in women at risk of HIV in sub-Saharan Africa. It is not a licensure trial, and this trial is being conducted in 5 African countries with just over 2,600 participants having been enrolled. So when you look at the Janssen's development plan, and they show us how they presented this at IAS last year, and they show how they move from the proof of concept study, the Invocado, to the phase 3 trial, the Mosaico trial, that is being conducted in the Americas and Europe. So you see the comparison of the regions for these 2 trials. The first looking predominantly at heterosexual women in a predominantly clade C region, and looking at intravaginal transmission, compared to the Mosaico trial that's looking at predominantly a clade B region in MSM and transgender populations, and looking at an intrarectal form of transmission. And the Mosaico study actually started in 2020. So the Mosaico trial, again, evaluating a similar regimen from the Invocado study with a few changes. It's a phase 3 trial enrolling 3,800 participants aged 18 to 60 years, and looking at cisgender men and transgender individuals who have sex with cisgender men and or transgender individuals. Bit of a mouthful there. And the goal is to enroll equal randomization across 2 groups, 1-to-1 randomization from the vaccine group and the placebo group. Again, 4 injection regimen that will be given over 12 months. The follow-up period for this trial is about 30 months. So you look at the Prepfact trial, which is another phase 2B trial. This one is funded by the EDCTP. This is looking at just over 1,600 participants in East and Southern Africa. The trial has already started. It started in end of 2020, and it's estimated to continue around 2023, but due to COVID, it may actually continue through to 2024. And it's looking at men who have sex with men, female sex workers, as well as general population individuals from Maputo and Mozambique. Also recruiting from Durban and South Africa, looking at the general population and female sex workers in Tanzania. When you look at the Prepfact study design, you see how they're evaluating 2 vaccine regimens, this DNA vaccine along with this protein that are given at 4 time points, as well as the second regimen that's again looking at this DNA vaccine, and again another GP140 protein that's given at similar time points. They're given at weeks 24 and 48. So what happens is in the first 26 weeks of the trial, participants will actually receive oral PrEP. They will receive either Descovy or Truvada. And this really, so you see that one of the goals in this study is in addition to looking at the efficacy of these 2 vaccine regimens, they will also be comparing the efficacy of Descovy relative to Truvada in reducing HIV incidence. And participants will be followed up for a period of time all the way through to about 2024. And in 2020, we saw the results from the HVTN 702, which unfortunately showed disappointing results of how the vaccine regimen that was tested in this trial was ineffective in preventing HIV. This was a follow-up to the RV144, which previously had shown efficacy to about 30%. Unfortunately, this particular regimen that was used in HVTN 704 was not found to be effective. So despite these disappointing results, I think it was of value to see how successfully they conducted the trial, and there were no safety concerns that were seen in the regimen. And the disappointing results of HVTN 702 continue to highlight the need for continuing the search for an HIV vaccine and looking further into some of the more recent and innovative vaccine approaches, which I'm going to talk about next. So in the next stage where we talk about these sort of products that are currently in early stages of testing, I will focus on just a few of them. There are many products that are in phase 2, phase 1, some in experimental medicine, but we'll focus on we'll just take a snapshot and talk about a few of these. And so if you look at the products and approaches that are in the early stages of clinical evaluation, there's a broad range of aspects that are being evaluated, starting from adjuvants and looking at various routes of administration, whether it's intramuscular administration or intranasal, mucosal routes. So all these are being looked at through experimental medicine trials, all the way down to looking at mosaic vaccines and vaccines that induce broadly neutralizing antibodies. So I'm just going to shine the spotlight on the work around vaccine development towards developing broadly neutralizing antibodies. So the field has identified this unique group of antibodies in a small proportion of chronically infected HIV individuals. And after years of infection, they have identified what are called these BNAbs, and these have been critical in sort of identifying where these BNAbs sort of bind to the envelope and being able to sort of map out these epitopes and looking at how these can potentially be designed into immunogens that can be administered through vaccination, with the hope that ultimately you'll be able to induce these BNAbs through vaccination. It's a very sort of, to put it simply, and we won't go into the details of process, a bit more complicated than that. At this stage, I think the field has not yet figured out how to induce broadly neutralizing antibodies, but in a series of trials that are planned over the next few years, we hope to understand how this may be done and whether this is actually feasible at all. So when you're looking at eliciting broadly neutralizing by vaccination, the fundamental goal is to be able to elicit sustained protective levels of BNAbs in people. And to be effective, the thinking is the vaccine will need to consistently induce BNAbs against multiple sites on the HIV envelope in most vaccine recipients. So one is not sufficient. So targeting multiple sites will be necessary to provide adequate coverage against the huge diversity of global isolates that an individual may come across, for example. So just to highlight some of the approaches that are being used towards BNAb-based HIV vaccine design, and these will be tested in a range of experimental medicine trials. So I will focus my attention on the germline targeting vaccine design, which I will talk about a bit more in the next few slides. But there are also there's also a lot of work that's ongoing in lineage-based vaccine design by other researchers and immunofocusing vaccine design. So just to highlight some of the germline targeting vaccines that came into the clinic and were being evaluated in 2020. The IAVI GO1 trial was looking at the EOD GT86 dimmer. and also the BG545 SOSEP GT1.1 also entered the clinic towards the end of the year. And I think this has heralded an exciting new era in HIV vaccine development, and some results will be shared at R4P by Bill Schiff on the EOD GT860 immunogen. So that's something to look out for as part of this conference. And I think the main aim here is to highlight and just give you a quick snapshot of the broad range of products and approaches that are in the early phase testing. Some of these BNAB guided approaches looking to induce BNABs eventually. And so the thinking is you will start off the immune system by a prime, and through sequential immunization with a series of boosts, that ultimately you'll be able to induce BNABs. And some of these approaches, as I mentioned, are being tested in the G001 trial, as well as the C101 trial, and also this study that's being conducted by the HVTN, looking at, I think, this a lineage-based approach. There are also native-like envelope trimers that are being evaluated, such as BG545 SOSEP. And this is different from the immunogen in that this is not engaging with the germline, but this is looking also just to induce neutralizing antibodies that have activity against tier 2 viruses. There are also other envelopes that are being evaluated by other groups in Europe and by Robin Shattuck and partners. There's also other work that's going on with viral vectors. The ChimPad, for example, that's been developed by Oxford University, that's will commence testing early this year. And this CMV vectored vaccine work done by Louis Picker, that's also going to be evaluated and just recently started. And also other important T-cell based vaccine approaches that are also being evaluated. In the adjuvant space, we do know that adjuvants have been used effectively to improve immunogenicity of certain vaccines, as well as being able to increase breadth of protection, and have been used as a tool for dose sparing, whether in reducing the dose of the antigen or the number of doses. And this is something that has been key, for example, in pandemic or epidemic preparedness. And so in the HIV space, there are several adjuvants that are being evaluated in multiple studies. And again, this is an exciting space with some work that's being done by Julie McElrath around the HVTN 133 study that's looking at a group of adjuvants, including the GLA, the CPG, 3M052 and alum, for example. And so we're all waiting to see the results of these. And also these are the additional adjuvants that are being deployed and utilized for other vaccines. And we'll also be seeing results from these in the coming years, months to years. So in conclusion, I do acknowledge that I didn't give you the full breadth of all the products that are currently in phase 1 testing, but just gave you a few highlights of some of the exciting approaches that are currently being evaluated. And so we do acknowledge that the HIV epidemic does continue to remain a significant global challenge, and an HIV vaccine is needed to achieve a sustained end to the epidemic. There are empirical vaccine approaches that are currently in efficacy testing, and 3 trials that are currently ongoing. There also is a range of exciting new vaccine approaches that are in early phases of development, including those that are looking to induce broadly neutralizing antibodies, those that are looking at native-like envelope trimers, that also could be part of the BNAB approach, as well as other T-cell-based and viral vector vaccine approaches that are in development, and some adjuvants that are also being evaluated in the field. And so I'd like to thank you at this point and acknowledge our funders within IAVI who support the work that we do, and some of the work that some of the vaccines that I reported are being supported by these donors, and also want to acknowledge the broader field that are contributing to this very rich pipeline of HIV vaccines and work that is promising, and we're looking towards the results that are coming out from these studies. Thank you very much.